Another thing about Canvas, if you have seen Canvas, hopefully you notice that there are folders in the file section. One right now says Exam 1 content. And then that is 2 folders, lecture slides and lecture reviews. What we're seeing today will be in the lecture slides section. After lecture is over, I will go back to my office and type out a few questions. Like, define this word, compare and contrast with these 2 things. Just to give you an idea of what did we cover that day. What are the types of things I expect you to be able to do with the information that was given in the lecture. You can think of them as study guides. I don't typically think about the lecture reviews as a true study guide. The questions on a lecture review won't be questions on the quizzes or the exam exactly, but the content will be the same. And one of the things I do try to do is change the way in which questions are asked from how the information was presented in lecture to see if you learned it or simply memorized this content. As an example, who knows the alphabet? I see some people don't raise their hands, well that means some people don't know the alphabet. But how early do we learn the alphabet? Pretty early, right? A, B, C, D, flip the song to make it memorized easily. So we all know the alphabet from A to Z. Who can tell me the 4th letter before P? What is it? How'd you figure that out? How'd you know that? But how would you figure it out? Would you just go A, B, C, D, E, F, G, count your way forward and then count your way backwards?
I kind of go way backwards for P by singing the song.
Yeah, so that's an idea of let's use something you learn one way and a different way. That was a simplistic idea, but it's a good thing because how many times do you think about the alphabet backwards? I have not been arrested or pulled over for a DUI or DUI, but I know one of the things they ask you to do is to say the alphabet backwards. Or start at a weird place and go forwards. And again, tests you of where can you go, where can you be, things. It's also sort of relevant for studying. Don't just study from the beginning of the section all the way to the end. Sometimes start in the middle, go forwards, go backwards. Any way that you can break up the information and change the content, give it your own spin to it, you're more likely to remember your idea on something rather than here's what I say, we should memorize this as the gospel. It's not the gospel, it's just here's some words that I put together on a slide. I'll try to help you as we go along. Like, what are some other things you can do to try to help? But let's just start with the basic idea. This class is called Introductory Genetics. And all the stuff with breeding is done already. So what do we have left? A lot of things these days, genetics means DNA. And so when looking at DNA, we follow into a category that's more akin to genomics. So there's an idea called molecular genetics, which is using DNA to study genetic problems. A lot of the things that we're trying to learn today don't have a one-to-one correlation. You can't make a mutant and you get a true phenotype. It doesn't happen a lot with humans. I'll give you a lot of examples where it takes changing multiple things to make something actually visibly different or measurably different. But we can look at DNA pretty easily, and that's what a lot of studies do these days. Does anybody know anyone that has undergone breast cancer screening or genetic screening for cancer? Do you know anything about what they did or how they did it? A lot of times they do is they take cell samples, isolate DNA, and sequence specific genes, look for specific mutations in those DNA sequences. And based off of that DNA sequence, they give you a probability or a likelihood of your chance of getting breast cancer. And sometimes it's high, sometimes it's low, depending on what type of mutations and what genes those mutations are present in. So DNA is the wave of the future. So it's important to talk about DNA. We spend a lot of time talking about DNA. I don't know if anybody knows about this particular genetic experiment. Anybody hear about the 2 twins that are astronauts? One went to space, one stayed on Earth. And they specifically found twins to do the study to see if they should basically be the same genetically speaking. Because they're twins. So the twins are, they're identical. These guys are identical. They sent one to space, or I don't remember whatever that means, I forget exactly. But they came back, they sequenced the genome of the one in space and one on Earth, and they noticed some differences. So the question was, what does extended exposure to space and zero gravity have on humans? There are some fun DNA changes, but we'll talk about those later as we get to those particular sections. That's kind of interesting, right? Your DNA does change when you go to space. It also changes back. What they found is that any changes that were a result of zero gravity actually reverted to be more or less normal. So even though they may change the space, it might turn back to be human. You may still human, but So, who wants to go to Mars and be actually different when you get there? Let's go, right? Be a Martian, because you'll be living on Mars. Anyways, some other studies, and all through the semester I will show things like this. And typically there are slides that you might not actually have in your slides, and this is an example of one of those. And they have this little symbol down at the bottom that means I'm going to talk about this, but you can check out, not pay attention, because I don't ever ask questions about these types of slides. I just like to talk sometimes and give you a break from writing things down furiously. There's some pretty fun stuff out here. Here's a study where they dug up a bunch of dead people in Europe and sequenced their DNA. And they found that there's this one gene, in particular they call it ERAP2. That's fun, right? Stupid infuriating people. Anyways, they found that certain people had a particular mutation, certain people didn't. It basically equated, if you had this mutation, you survived the Black Plague, and you were not going to get the bubonic plague. If you did have a mutation, you would, or it was reversed, it doesn't really matter. But these are things we see a lot, where a particular mutation shows up. It does give a phenotype you wouldn't just measure, wouldn't go around and say, can you survive the plague? It's not red hair, green eyes, things like that. But still, it's an important phenotype that you can have based off of your DNA sequence. And by looking at DNA of different organisms, some people think that we could turn back the biological clock. Can we de-age animals or plants? Anybody know the effect of longer life? Think about in the 1900s versus now, what was the average length of time for human life? What was the age expectancy or life expectancy?
It was lower than
Yeah, it was a lot lower. Even in the 1800s, you have people not living very long. 30, 50 years, then it was 70 years, now it's 90 years. So what is the implication of living longer? I'm going to tell you one of those implications is more mutations accumulate over time. So can we change that, effect? And there's one study where they tried to turn old mice young again, and I guess they succeeded because they said, yes, you can. But what would that look like? What would it look like if you de-aged humans? What would it look like now that I'm 100 years old? What would it look like? Have you ever seen the movie In Time, I think it's what's called, with Justin Timberlake and what's her name? Amanda Seyfried. Basically, people stop aging at age 25. So, cool. Would you like to live forever? Would it be worth it? It's another philosophical question. But morality is not really that important sometimes in science. Sometimes we do without thinking, and then the question is, what should we do with what we've done? Which is the problem with a lot of things. Anyways, one thing we will spend a lot of time on this semester, hopefully, is the idea of genotype versus phenotype. What does one thing represent? What does the other thing represent? And one of the more important things about genotype is that it does refer to your DNA sequence. Those A's, T's, G's, and Z's, how are they put together relative to somebody else? And we can learn about people's propensity for disease or likelihood of certain types of athletic ability through DNA sequence. And it's not until like the last 30 or 40 years that being able to use DNA technology has become widespread. That's why you can poop in a box and send it to a company. They'll tell you your chance of getting colon cancer. You can do a cheek swab, and send your saliva to 23andMe or Ancestry.com. They can tell you something about you. So a lot of companies are doing these types of things. But genotype is something that is in your DNA. And it can be changed. We'll talk about different ways that can affect the genotype of an individual. But typically we see the manifestation of the genotype in the phenotype. What do you look like? What kind of characteristics do you have? And we call these typically physical characteristics or outward traits, but some of the phenotypes that we're going to talk about are actually in the DNA sequence. So things you can't really see. Your blood type is a phenotype of you, but can you see that when you look at a person? And so there's things that we can know about us based on our DNA sequence that we don't actually see in the phenotype. Now historically, phenotype was what we used. Charles Darwin went around the world looking at different things and saying, you look like this. You look similar. Maybe you're related to each other somewhere back in history. Who decided that the lions and tigers were two different species? Somebody thought they were different. Why is a greyhound the same species as a chihuahua? They don't look that similar. You don't know those are two different dog breeds. The greyhound, not the bus. The dog can show you something else. Rather greyhound. Who thought bus? Nobody thought bus? Maybe Greyhound buses are not very common anymore. But this, Amtrak. Does anybody know what Amtrak is? Okay, it's like a phone booth. Something that's disappear over time. Anyway, that's not really important part of this class. But what does a dog breed? What does a cat breed? Are humans breeds? What are the different races of humans? Is it akin to the cat breeds or dog breeds, or is it something different? It's a philosophical question, but one thing you might want to think about in terms of genetics, what makes something different? Sometimes an arbitrary distinction when you think about it. But they'll tell you that humans are all 99.99% identical to each other on the DNA level. It's a fact. We can see it. So small places of differences are there. It gives a lot of differences in phenotypes. Height, weight, hair color, skin color, eye color, all things can be changed. So as we think about what affects a phenotype, there are two things we need to think about. It's not just a gene that can be affected. Sometimes gene expression can be affected. If you remember anything about biology 140 and 130, there are two important processes of cells will form called transcription and translation, where we use DNA to make RNA as the first step, then use that RNA to make proteins as the second step. So we transcribe DNA to make RNA and translate RNA to make proteins. And so we see the information contained in DNA physically turned into a protein molecule. So whatever's in our genotype is going to be a phenotype. There's a direct comparison or connection between genotype and phenotype we want to always think about. DNA changes can cause protein changes, and protein changes can lead to phenotype changes. Sometimes the phenotype change is because of a mutation in a gene, causing the protein to act differently than it did originally. Other times, the DNA change we're going to see is going to affect how much of the protein is produced. And that can also have a profound effect on a phenotype. Make a lot of it, maybe, let's say one color, make a lot of less of it, maybe a different color. It's actually one of the things that's interesting about both eye color and skin color. The amount of something you make has an impact on the shade of your eyes or the shade of your skin. If even hair color, all in the same category, which is what gives us a lot of different variations in those three traits. It's not just one or the other, it's a gradient from one side to the other. We have names that classify these things, and we're not gonna kill them today, but I'm gonna spend a lot of time talking about this part right here, not so much about this one exactly. We can change the protein function, or we can change the protein amount. Both will have an impact on phenotype. And both are due to typically changes to the DNA sequence in some way or another. And one of the other important things to do is make sure that we're using our terminology correctly. And a lot of times this falls into trait versus phenotype. So I've seen this several times in the past, so adding that slide so we can try to deal with sort of chicken and egg type of idea. What's the overarching category and what is a subset of that category? And so trait is the big umbrella thing, and we have lots of phenotypes for certain traits. So just pick one, for example, eye color, green, brown, blue, are not in all of the eye color possibilities. There's some of the more common ones. So these are our phenotypes of a particular trait. And for blood type would be the trait, and the actual blood type would be the phenotype associated with that trait. Let's go with skin color, hair color, height, weight, all sorts of things that we can think about. Stripes versus spots. Ever think about how do you go from DNA to making spots on an animal? Or stripes, when you think about that. I think it's weird. I actually think it's so magical, but it also works. But traits versus phenotypes, we'll try to make sure that and that's me, mostly when I say trait, hopefully I'm using it correctly and we all know that that's the umbrella term. And a trait can have multiple phenotypes, not the other way around. When it comes to phenotypes, there are two that are particularly important: wild type versus mutant. We define the wild type as the most common phenotype of an organism. So let's do a little bit of an class assignment. What are wild type traits for humans? And so what do you really need to be wild type? What do you think? What is the most important thing that defines what wild type would be? Most common doesn't really help us, right? What's that? I'm not sure that's where I'm going with it, but you're on the right track. What do all humans have? It's kind of where we can go with thinking about what is the wild type of humans. We have hair, we are hairy people, or hairy beings. Is that the only thing that defines a human as a wild type? That we're hairy?
Melanin.
And now we got a lot of things. So we have 2 arms, 2 legs. So, we're bipedal. I'll just said something else.
I said melanin.
Melanin. Yeah, okay. Let's see, how do we think about melanin? I mean, we all produce melanin, yes. I just want to think of this, like we're trying to define what is a human. If you describe a human to an alien, is this cat a human? Is this dog a human? Is this snake a human? What makes it human a human? And so think of what do all humans have? 2 eyes, 2 ears, 1 nose, types of things. And so for wild type, human doesn't tell us, or wild type for human doesn't really tell us a lot. But sometimes wild type for bacteria, for example, can tell us quite a bit. Because we assume certain traits within that particular world. And some terminology between bacteria and eukaryotic organisms and humans doesn't always translate very well. When talking about bacteria and prokaryotic organisms, the wild type is very important. But we want to know what is the usual phenotype of this organism. And maybe there isn't a wild type for humans, or dogs, or cats, or tigers, or lions. Maybe there is. But something I'll never ask you to do is sometimes interesting exercise. So if we define something as the norm, anything that's outside the norm becomes a mutant. That's where we have to be sort of, what's the word? Careful, I guess is a good word. Because what do you define as being normal? Is a certain height normal? Is a certain hair color normal? Get into weird ideas of discrimination. And we want to keep that out of genetics as much as we can. But when we define something, what is normal? Typically in the world of molecular genetics, it is what is least likely to give or cause a negative phenotype, sort of in a normal state. Anything that is more likely to treat or lead to a disease or a change in phenotype might be considered something that would classify as a mutant, something that is needing to be thought about as going to be something different. And not all mutants are bad. Sometimes they have superpowers, fight crime. At a certain point in evolutionary history, we have the same eye color, so a mutation happened. And now we have 2 eye colors, and another mutation, we have 3 eye colors. Another mutation, now we have lots of eye colors. So, mutations aren't bad on their surface. Depends on the type of mutation, we'll talk about that as we go along. I'm using a word a lot, but I don't know if you remember what it is, but the word is gene. Does anybody remember a definition of gene from Biology 140? Sorry, what'd you say? I didn't get everything that I want. Yeah.
A section of a chromosome?
That's getting closer. So we define a gene as a section of chromosome. And we have that in your little part about that encodes typically a protein. That's one of the most common examples or definitions of a gene, a chunk of DNA that is used to code for a protein. There are some differences and extensions that we can add to this. One of the other more common ones that is sometimes important to think about is sometimes genes don't code for proteins. They code for something called a functional RNA. And so to go from DNA to protein, we need both transcription and translation. Sometimes DNA sequences are only transcribed and not translated. So we do see that there are some what we call functional RNAs, like the messenger RNA, the transfer RNA, not the messenger RNA, the transfer RNA and the ribosomal RNA will be examples of functional RNAs. They're not translated. They function as RNAs. And in this class, we get to hear about a lot of other types of RNA molecules because they control gene expression. And again, gene expression is the transcription process and the translation process, going from DNA to protein, what we call gene expression. So we have a gene that has a certain DNA sequence, and if we have that same gene with a different DNA sequence, we call that an allele. And almost all genes have alleles. Sometimes there's lots of alleles, sometimes there's very few, sometimes they're dominant, sometimes they're recessive, sometimes they're neither. So fun categories of things we can talk about. But alleles give us the differences that we can think about. A lot of genetics is trying to identify what alleles are out there in the world, and is this allele a good allele, a bad allele, or a medium allele? Sometimes there are intermediate states of being good or bad. And sometimes dominance and recessive are not really easily identified and classified, but we can think about dominance and recessive quite a bit. Now the other thing I think is interesting to think about, wild type doesn't necessarily mean dominant. A lot of people sort of equate those two things together. Dominant is not always the most common trait out there. Sometimes recessive is the most common trait out there. There are some disorders that are called autosomal dominant or X-linked dominant. And there are other disorders that are called autosomal recessive or X-linked recessive. And we do see that sometimes a recessive allele shows up more in population when you have an autosomal disorder or a dominant disorder. And reverse is also true. If the recessive disorder is in the population or is a disease, sometimes you see a lot more of the dominant trait or allele in the population. So you sort of weed out, not really weed out, like intentionally weed out, but sometimes disorders cause the individual to not be able to reproduce as efficiently as another one. So you might die before you have children. That's sort of nature's way of weeding out undesirable traits because they're not really able to survive on their own. That's the other thing we're seeing in the world of medicine these days. If we're living longer and medicine is able to treat a lot of the things that would normally kill you 50 years ago, you see a lot of sort of differences in how traits are sort of talked about nowadays. I see everything in the population now because there's nothing weeding those individuals out of the population through natural death. We can cure them or treat them, keep them alive for a lot longer. So, increased life and good medicine does have a drawback. It does sort of make lethal or negative alleles show up a lot more in the population. But genes and alleles, gene is the main one, and then the alleles are the different ones, the variants or different versions of the same gene. It's really important because sometimes we think about alleles as being different genes. If it's a different gene, then it makes a different protein. It's a different function. Well, same function, but altered function. It's supposed to do the same thing, but now it's something slightly different. You may know what the most common cause of mutations or changes in DNA is from. Do you have any guesses, thoughts, wild ideas?
UV radiation.
You know, got UV radiation, anything else want to throw one out there? Any guess? What's any ideas, thoughts? Any alive? As they say, anybody respiring out there? If you're alive, then your cells are undergoing aerobic respiration. The joke is, are you making carbon dioxide? Then yes, you're alive. Radiation is not the most common cause of mutations. A lot of mutations are caused through errors when we copy DNA. That's where a lot of our changes naturally happen. And so we have this natural system where some mutations don't get fixed. And it's nice to leave some mutations in. Can't think of why we want to have mutations out there in the population. We want to have people or individuals with different traits.
There's advantageous mutations.
Okay, so, give an example or just, oh yeah, not to be me, I just wonder if you have an example.
Thinking.
Does anybody know the answer to this question? What happens if everybody is exactly the same?
One disease can wipe everybody out.
And so that's the one problem with genetic homogeneity, is a single disease or disorder is going to affect everybody exactly the same. If that's death, everybody dies. But if you have mutants in the population, some of those will survive, and the species still exists. So every once in a while we tweak things to see is this better or is this worse? That's sort of the idea of evolution and genetics, is make a change, sort of do the experiment, see what happens. Is it better or is it worse? Is it the same, And sort of life chooses what is sort of better by surviving better, being faster or stealthier than somebody else. Not really genetic pressure on humans like there are other animals. But still we have replication errors is a really important driving factor for making mutations. The other ones are exposure to radiation and chemicals. So UV is in that category, X-rays, gamma rays, things like, asbestos, nicotine, all sorts of things can be cancer-causing agents. But some that you probably never heard about, maybe you heard about this one, 140, but your DNA moves itself a lot. Sometimes that can make mistakes. And there's also DNA in you that moves itself, which can also cause mistakes. So there's all sorts of fun things that happen with DNA. Lots of things can make DNA change. When we make mutations, And we can either change the functionality of the protein, or we can affect the expression of that protein. So we can either make the protein function better or worse or the same, or we can make more of it or less of it or the same amount of it as we did previously. And again, that can have either profound or not so profound effect on the phenotype that we see for an individual. Given gene expression, transcription and translation going from DNA to protein. So we're going to see this slide several times this semester, which will say these are the things that can cause mutations. And we'll go over some of the more important, or just a few examples of chemicals that cause mutations, what mutations they cause, and how we fix them. So we're not to that point yet. The beginnings of walking before we run. And one more thing to sort of think about. Mostly what we hear about in the world of introductory genetics are the idea of simple traits. Simple traits are easily identified and explained. But unfortunately, most of the things that affect humans are actually complex traits. So when we define a simple trait, we say that it is affected by a single gene. And very rarely is this the case. We think of things simply, but a lot of things are actually much more complex, and that's where we spend a lot of our time, on these complex things. But simple trait is something that is only controlled or affected by a single gene. Complex traits are things that are affected by multiple genes or also environmental factors. And we don't spend a whole lot of time talking about environmental factors and how they affect things. But typically that falls into the category of exposure to chemicals or radiation, which can cause mutations. Some examples, again, skin color, eye color, hair color, are ones we'll talk about quite a bit in this class. We know a little more about those things. Blood type is one thing we'll talk about as an example with dimples. I'll talk about dimples. I just had to find another example, and that's what Google told me, dimples. Simple traits and complex traits. All sorts of fun things. Now, if you're really interested in humans, you can go to this website called OMIM. As you can see, OMIM doesn't stand for anything that's on this slide, because I forget what it stands for. This is not an online catalog of human genes and genetic disorders, because MIM is not that stuff. But if you're interested, and I tried to do this website, it sucks. It's not helpful whatsoever. You actually really need to know something to actually use this website. But it's not really for the lay person. But one of the things I did find on this website is this one slide I thought was fun. It falls into the category of simple traits versus complex traits. And what they are saying is that there are 3,446 genes in the human genome that only affect one phenotype. And there are 251 that affect 4 or more different phenotypes. Imagine a gene having its hand in 10 different puddles, saying, I get to make you blue or you green, but you have red hair, curly hair, and all different types of things. Sort of a weird case. Some things do multiple things in the genome. And we're trying to figure out, like, what are these things, what do they do, and how important are they? It takes time, and we just need more DNA sequence evidence to figure out what these things are. But simple gene disorders and traits, 4,559 genes when it comes to complex traits, a lot less. So maybe there are more simple traits out there than we actually think. But it is a definition you want to understand. Simple trait versus complex traits. We go beyond just simply complete dominance, incomplete dominance, and codominance. There's a lot of other fun things to think about. this next slide is gonna be an introduced topic that is gonna take more than 6 minutes to go over. So I'll have to talk faster than I normally talk. Usually I let people go early when I pay the price. Later in the semester, if I let people go early, I have to talk faster, because I don't have less time. We'll actually do this on Wednesday. Lots of ways to talk about a trait, not just whether it's a simple trait or a complex trait. What does the trait actually do, and how do we measure that trait? So we'll talk about morphological and molecular quite a bit this semester, but also say a little bit about this physiological versus behavioral traits, and what we can do with these things. So on Wednesday, we'll dive directly into how do we look at a morphological and a molecular trait for the same gene, and how do we think about things. It's a little different. So, do we have any general questions about today? Specific questions about today? Anything done for the day? I have one more class today. Don't get too hot out there.